The diagnostic lab in Iowa is actually housed at the College of Veterinary Medicine. They're in Ames. And it's a large college because Iowa has a contract now, I think it's with 5 different states. And so about half of the students are from Iowa, and then the other are via contract, and then a few out at large. Within the college, we're housed within a department that's called the Veterinary Diagnostic and Production Animal Medicine. And that group is made up of 2 large groups. It's the Food Supply Veterinary Medicine group. And so those are the people that do most of the teaching, or primarily all of the teaching of the professional students for the food animal curriculum. And then there's our group here at the diagnostic lab. The diagnostic lab is a team that's about 22 faculty and about 100 technical staff right there at the college. And we do an awful lot of this every day. Probably about 90 to 95% of what we get comes in a box. Pieces, parts, fluids, a little over 1,000 times each week. We run it through the mill and spit out the results to our clients. We're somewhat unique as diagnostics labs go because we're so heavily dependent and focused on swine. We're about 75% of all of the diagnostics work that we do is swine, the other 10% bovine and 10% poultry. And in Iowa, the poultry industry is largely layers, as well as some turkeys. And that's kind of in part because of kind of where we are relative to where the pigs are. And then because of the network that Iowa has, there's actually a little over 500,000 pigs, believe it or not, that come into the state of Iowa every week to be fed. And so we have a lot of ties to our surrounding states. And another very significant driver is really these tremendous systems of logistics that we enjoy has really concentrated the swine diagnostic world to a few institutions that have put a fair bit of capital into it. This just kind of gives you a little bit of a feel for where our cases come from. So last year we had about 70,000 cases. Right at half of them came from the state of Iowa, and then the others from around. And so where we are here, you can see serving in the state of North Carolina, and the pork industry in particular is a very important part of our business and service. And it's a very, from our view, a very good ally and partner in helping support the veterinarians and producers in the state of North Carolina. And so about 9% of what we do comes from the state of North Carolina. Whoop. So now just get into the PEDv. Many folks have seen this, and this is really just kind of the, again, not number of sites, but just number of cases that have been observed over time and how that's grown, where we were a year ago about this time to where we are now. So really a pretty natural kind of inflection point curve as it went across the country. This is what that number looks like by week. And so you can see once it kind of picked up steam, as soon as it started to get cold and really got in the heart of the Midwest, the numbers really took off, and then have since, when it started warming up, have declined fairly precipitously. And and this is kind of where that number looks like as far as distribution cumulative to date across the U.S. And I think the one thing what's really clear to me, or would appear, that this distribution looks very similar to this graph. So believe it or not, the disease is where the pigs are, right? And found its way across the U.S. In very short order. I think a little bit of good news that kind of goes along with this warmer weather, and then a lot of herds that have been recovering since they broke in the winter. The percentage of cases that are testing positive has dropped from about 35% to just over 10% here over the last 4 months. The other thing, quick, a little bit about PDCOV. We got so many of these acronyms now. It's like, my God, there's only so many letters, right? And it's confusing. You know, what is this? Is this another strain? We got strains of PDv, and now we're talking about this thing called Delta coronavirus. And the one thing I just wanted to remind folks is that this virus is, from a genetic standpoint, is quite different. It's very different. So it's a coronavirus, causes similar disease based on clinical signs being reported from the practitioners and producers in the field. Maybe just not quite as severe, but it's quite different than this other family. So it's kind of in its own clade. It's called a Delta coronavirus, where these other viruses, meaning PDv, TG, and PRCV, are up all in something that's called an alpha coronavirus. So again, the main thing there is that the Delta coronavirus is quite different. And the other thing, that the current state of that from a diagnostic standpoint is also quite different, meaning really the only tool that we have to date is a PCR for detection. And we can sequence it, but it's not being done routinely. It's more just whole genome for kind of research purposes. And to date, there's no serological test available for that disease. And this just the same thing for Delta coronavirus, kind of what that curve has looked like. And it was definitely quite a bit later to come to the party. And the reason this started going up here is, well, that's when kind of got a first case or two, and actually we had to then develop the diagnostic test, and then guess what? You start finding stuff, right? And but it's the main thing here is we're talking hundreds, not thousands. Okay, if you remember, the last graph was 7,500, 8,000, and this is 400 to 500. Okay, so quite different, and that this is what this looks like by week. So once again, it kind of was here in the middle of the spring, and then it's gone down as it's gotten weather. But again, probably not near as much testing and so on going on before because there wasn't a test available. And the reason that all came about is we had these kind of hit and miss cases coming through that were the practitioner was saying, boy, it looks like PEDv, but not quite as bad, and we're doing all these tests and they're all negative. So what's going on? And that's really ultimately what was the derivation. And so we just finished here just recently. Us and some of our peer diagnostic labs that do a fair bit of swine work went back and tested the freezers again and see what we could find. And we found actually the first case at our lab was back in August. We found that it was just a couple sites in Iowa. And then between then and now, once this got going, there really wasn't that much. A case or two, I think one was in October, right? So it was around, but not too far. But we tested thousands of samples prior to this, all the way back to December 2012. So we're very confident that it wasn't here prior to the PEDv business. I said fairly confident, right, that it wasn't here, and definitely hasn't maybe quite spread so much. The other thing I think that's kind of unique about this is the distribution of this is quite different as far as across the country. If you look at just kind of where this is at, here, for whatever set of reasons, it's kind of north and east, and again, not so much as much out here. So a little bit different distribution than the way PEDv took across. PCR has been the primary tool that's being used and kind of been carrying the weight. Came on very quickly, very sensitive. This is for the guys in the room. It's not, more isn't always better, okay, in this world. From a testing, because the feces cause inhibition, okay? So, and it was crazy, especially when we first got into it. It's like, what does they say? Mother's necessity of all this something. Is when we first started getting this, I mean, we started getting bags of feces, right? I mean, like God, oh, how much do we need? Okay, send a lot, right? It's like, so we're getting these bags of feces, and then the gals in the lab, okay, now we actually got to test this stuff, right? And it's like you get this little wood, those little wood sticks, okay? It's just like a little drop of that in a ml of water, or in a ml of PBS, and that's it. So we don't need that much. So it's kind of evolved to a lot of fecal swabs and oral fluids being tested. Just a little bit about what the virus, these coronaviruses look like, and what the PCRs are based on is one of these two genes, depending on the pathogen and the specific assay. But they're based on what's called the nucleocapsid protein here, or the N gene, and the matrix or membrane protein. And these are more conserved genes, okay, so the nucleocapsid and the membrane protein. Whereas the sequencing, we'll talk about that later, they're based on the more variable genes, okay, because when you want to just find it, you're looking for the most conserved target, okay. One thing that's kind of been surprising, and actually it was a lot of the really early work that occurred out here. I think it was Emily did a lot of this work early on, and this was quite surprising to us actually, in that this oral fluid is really holding up as being a fantastic detection tool for these growing pigs for this fecal oral disease. Historically, we've always thought about it as far as for systemic disease and so on, and we had known for some time with PERS that it's truly an environmental sample, okay, and that's what makes it so sensitive.
because the pigs are always sampling the environment, and then they chew on the rope, and when we find what's on the rope. But it's really came out that really this is really a highly effective tool for these fecal-borne diseases, and it's quickly become the kind of the tool of choice. The other thing is we get a lot of these different kinds of swabs in, and this is mainly where people are screening wean pig populations to say, have they recovered, are they still shedding or not? And the other thing that's really come out of this is we get a lot of these kinds of swabs. They're kind of like this. They come this, and they like have a little sponge on the bottom, right, and all that. And what we really like, and this will be something we're kind of putting a little education campaign out, is what we'd really like is these Copan swabs, okay? And because they're these flock swabs, and then they're actually put in media, okay? Because we actually test liquid, right? Does that make sense? And so like what happens now with these things is we get them in, and then we have to take these, and then we put them in like a mill of PBS and kind of stir it around, okay? Put it back. So we get one of those, and then we get pool them in 5, right? So you do that 5 times, and then you take a little bit of each one of those into another tube, right? And with these is the same kind of things. You just stick these in, they break off, you screw the lid on, then this is, and here you go, right? And so it can be much more automated. So as we transition with this, there'll be a big push to say, how do we get more of these kinds of swabs, just to help streamline the process from beginning to end. And it's actually going to be more sensitive too, because rather than relying on us doing this, right, thousands of times a day, is this thing's been shipped overnight, shake it in there, released into the fluid, and so on. So it'll be a better sample too. And again, those are just kind of, I think, just simple but practical things we learn as we go. This group here is our molecular diagnostics section. They've been a busy group over the last year, both in doing the work, just for this particular pathogen, PEDv, just to show how the increase in demand for that assay has been being used by the industry, a little over 10,000 a month now. But it's also been a lot of innovation that's been required as we've got into this. And so the contrast doesn't show up so much. But these red, basically all of these, those say would be all new assays within the last year, again, that were developed. So both screening PCRs for PEDv and the Delta coronavirus, as well as we have a differential PCR that differentiates between what we call the original strain versus this variant, or an Indel strain that some are calling it as well, to differentiate between those two strains, as well as the sequencing assays. Again, the sequencing assays are based on the S gene or the spike protein, and the reason that is because it's much more highly variable. Okay. Again, that's being done routinely. What we're doing is what's called S1. Again, it's the most highly variable part of the S gene. And one of the things that's come out of that is that's really where we weren't doing a lot of sequencing. Really, it wasn't until, I can't remember when it was it the first of the year? Something like that? We really weren't doing much sequencing because there was some done early on, but they were all the same. So it was like, why do it? And then like most things, it comes from the practitioner saying, hey, I got this thing. Again, doesn't look like other PEDVs that I've seen. Doesn't make sense to me, all that. And they said, why don't you sequence these? Okay, and it was really through that work that we found, oh, geez, there is a different strain. And it's quite different, right, than the first one. And it was about 6% different using this S1 gene as a variable. And one of the things that kind of was confusing early on is because when you do the whole S gene, which Minnesota was doing at that time, it was only 3 to 4. Whether it was the same, well, yeah, they're the same. So 2 strains. And so the basic message is, yeah, there's 2 strains in the U.S., little variants within them, but 2 primary core clusters. And then in retrospect, so we went back and tested again. So we went back to the freezers again and said, okay, how long has this thing been here? Okay, and lo and behold, we found it in May 15th, I think it was, of 2013, right? And the very first PEDv was found when we went back into the freezers and did those retrospectively was April 15. Okay, so I'm sure that's more within the margin of error of an introduction, if that makes sense. So whether it was exactly the same or not, it was pretty much in and around the same time. Serology. Serology, one thing I've learned about this program is
PCR technology and developing PCRs is a much more straightforward plan than getting a good serology tool in place, okay? We've had some luck, not quite as fast, but we've had some luck with the PDV, and really that came about through traditional technology, but the core to be able to do that was you had to grow the virus. Okay, so we had to be able to cultivate and grow the virus to get this IFA assay. And that's really, I mean, really held up it's robust, it's kind of stood the test of time. It's very labor intensive, it's subjective, it's one person on a microscope looking at one well at a time, this kind of thing. And so there's been efforts to try to get some ELISAs, some based on proteins and so on, and then and those have been definitely okay, but they don't last very long. The immune system, it goes away so fast that it's really a fairly short window. And so what we've been working with is, and just getting ready to launch this, is what's called a whole virus ELISA. And basically what it is, it's an IFA in a PDV format, or excuse me, it's an IFA in a ELISA format, if that makes sense. So it's coding the cells, the whole cells on the plates, and that's why it's taken us a while to get that up and going, because you have to cultivate massive amount of virus and code the plates and find a way to do that consistently. But this thing is looking good, I would say. And one thing we've learned about once we've started doing some serology, especially for, this is a practical thing, especially for gilt grow-outs, as we work with a fair bit of people that have multipliers and so on, it's really clear that these healthy gilts, you can if you want to say, are they really naive? If that was your goal, you can miss it, okay? Because we've had a number of these cases where a breeding stock company said, well, there's no way these are positive, right? Because they'll do a serology at the end, right? And so clinically, in these really healthy growing pigs, to say that it can be readily missed if naive females is your concern. Okay, so that's kind of so basically it's like we got some IFAs robust. This will be new very soon. SDSU has a good FFN, a very good test too, I think, for people to look at and consider. We have not been able to bring that technology on. And then again, there's some folks working on some developmental stuff for Delta, but nothing available that I'm aware of to date. The IFA has been what we've been doing. Comes on 3 to 4 weeks post-exposure. Challenge is the titer doesn't last all that long. Even with this, you're out at, say, 6 to 8 weeks. This whole virus ELISA will be new. The upside of that is it is more sensitive, okay? And it's gonna last longer. So we'll get a little longer antibody titer detection. And the other thing is it gets us in a format that we can modify for milk and colostrum evaluation as well. Okay. The downside is these antibody assays today are pretty much, evidence of have they seen it, this kind of thing. We really don't have any feel at all to say, well, how does those results relate to any kind of protection or immunity? All they are really to date is an indication of previous exposure. Immunohistochemistry is fine if it's there, but because once these animals are infected, they lose their villi so fast, it's an incredibly insensitive test. Okay, so it's very meaningful if it's positive, but can quickly go negative because the villi they infect are actually gone. VI is very time-consuming and very low success rate, so not a very practical tool for routine diagnostics. And today we do not have an IHC nor success with growing the Delta coronavirus. And just kind of a picture of that. So this is a healthy gut. This is an acutely affected neonate, basically, and that's just stained with a monoclonal antibody, right? So basically, it's like the whole gut is coated with that virus, but then they quickly slough, and so then essentially that's why that test has been a valuable tool, but surely not very sensitive. Okay, so questions on the diagnostics. I'm going to go into a little bit of what we're doing more on the diagnostic information side. Okay, but I wanted just to stop here and see if there's any questions on the diagnostic assays themselves, or kind of the status of where that's at. Okay. Well, it must have been really, really boring, or it's crystal clear, right? Let's go with the lab. I don't know. Who knows? So anyway, and there's been every bit as much of energies on the back side of this around this in this arena as there has been on the assays themselves. And this is really what we're trying to do. Is how do we improve the quality of our information? How do we improve the value of the diagnostic information, its utility, its connectivity, its convenience for the user, and really enhancing our systems of traceability by what we've learned by some of this PEDv business. But again, the core things are quality as it relates to identity preservation, value, and utility. And so we started this kind of effort in 2013, and it's like anything with IT, it just doesn't happen as fast as you'd like to think it. It doesn't always work, you know. But these people are smart, but it isn't easy. So what we did started really the development and planning and getting organized and really getting those guys engaged in it to say in 2013, and then as these developments have been rolling out in 2014, and it's kind of been a stepwise plan with a common end goal. And so what we try to do is well, let's redo our submission forms, and really what that means, it's the data capture plate. So what data are you capturing on the information on your submissions? Okay, and the biggest thing we wanted to do is separate owners and sites, okay, and get more site, premise, flow, group, lot type information, ability to be captured in a standard way, okay. And also we have some folks, especially with the advent of all these sow co-ops, of people that want to affiliate the results with somebody else, okay? Because I'm an owner in a sow farm, I want to see my sow farm results, right? But I'm in another state, but I want to see does that make sense? And vice versa, meaning the people that have the sows, they want to see what their growing pigs are doing, okay? And so affiliates are just a way of sharing the results with who the submitter wants to share them with, okay? And that's okay. So that's to say, well, what additional data do you want to capture? And then, but then you got to say, okay, we got to redo our LIMS system to be able to capture and report it. Okay, getting it on paper doesn't do anybody any good very long, right? So you got to be able to capture it and report it. So we had to update our LIMS system to be able to capture the additional and report the additional information. And another key part of that some functionality we wanted to build into it is we wanted to be able to pre-populate our LIMS system with our clients' sites. Okay, so we're not subjective to what somebody types in and what somebody calls something, because it's never ever the same, right? And so the idea there is to say, well, let's set up a system where people can, but every time we get a download from somebody, say we download it out of our system, they send it to us on a spreadsheet, it's got all the site names, prem IDs, all that. Oh, it's always right? Okay, and so we wanted to be able to say, how do we leverage that? And then the other thing is we wanted to be able to create the ability of, if I had a group of sites or a list of sites that I want to affiliate with this entity or that entity routinely, that could be done on a site level and not case by case. So I don't have to remember to do it every time I submit it. I can say, here's the sites I want affiliated with this group, and it's done. Okay? And then, then be able to update the web reporting tool so it can report the additional information, and then get an improved web submission tool that basically resembles all of the above. And so this was kind of where we came out with two things. There's some species-specific stuff, but the core differences here, and I'm sure you can't read that, but the core differences were really here at the site level. So before we just had owners and then reference, and then that wasn't very functional. And so it was like, okay, let's separate sites from owners. We added a thing called division as well. So owners that have multiple divisions, we can divide it by division. Another way, it's mainly ways to cut the data. How do you enhance people's ability to cut the data the way they want to see it, right? And so we have sites separate from owners, and then this premise ID thing, we'll talk about that a little bit. We wanted to incorporate that, make that a little more prominent, and then get people for lot group, source flow, and then still miscellaneous reference. And we also wanted to have the ability to capture and report vaccination information to help make this thing that if there's any sort of programmed disease that evolves that has vaccination as a component, that we have the ability, again, to be able to capture and report that. Okay? And then some improved things with our test selection format, more modernizing it to the molecular era. And then the other thing is that's really changed with the molecular diagnostics is the range of sample types we get it's big. All kinds. It's like, you send it, we'll test it. And the challenge is that a lot of those different sample types have different techniques that have to be done to them. Does that make sense? Different extraction techniques and so on. So it's important, believe it or not, that we actually know what the sample type is. Okay? So we added the ability to do that. And then did the same thing for the path form, same idea. And this is really a key part I mentioned earlier, is this pre-population. And so this is where we've just started to reach out now to some of our larger clients to say, let's get this done. We'll send you a spreadsheet. You send us all your sites back. We'll upload it into our LIMS system. Okay? And what that does is, so that way when it comes, like push comes to shove and we get the case and we got this piece of paper and we got to enter it into our LIMS system, right, accession it, it's like, okay, we got a veterinarian, he's associated with the clinic, and then he's got a site name, okay? And for every folks that have a number, we'd like to start this, if they have a number that's associated with the site, we'd like to use the number first, then the name, okay? So when our people start to type, the numbers are a lot more repeatable than names. Does that make sense? So just a real simple system. And then what happens is so when they get to here, this is the case entry screen that the clerical staff is seeing when they're going to accession the case. If they've been preloaded, then it's go to this thing up here that's called preferred, and it'll have all the sites just for that owner, okay, in some sort of order. And it's kind of a type ahead. So in this case, when he started typing 6137, that pops up. They click on it, and it populates that, and then pulls all the correct information with that site up into it. Does that make sense? So it's a way of really enhancing the whole identity preservation of preservation of the information. Okay. Then you've heard us talk a little bit about these prem IDs and all that kind of thing. That's great. It plays all into this thing too. In that if people that are preloaded and then they use these prem ID stickers, the upshot of that is once all we have to do is scan it. And then it just pulls the information all in. Does that make sense? So going back to this whole, how do you reduce chances for error and make it more streamlined and repeatable? And so the Prem ID sticker thing plays into the pre-population things, because then it's just a scan. It self-populates, and we're good to go. And then if folks want these barcoded Prem ID stickers, we're providing those. Again, basically things on the website. You can download, or we'll send you a spreadsheet. You send it back, and we'll pre-populate the LIMS. So those things are all in place and moving. This is the thing that we're actually just started beta testing with a couple clients just this week, is our web submission tool, new web submission tool. And basically it was built to resemble the new forms, the new reports, just looks exactly the same. And the whole goal around the web submission bit was it looks exactly like the paper on both ends, for the user as well as at the lab when it prints out. Okay? And so, and the idea there is, okay, the practitioner, they log in as themselves, and then they have to have pre-populated the sites for their sites to show up. Okay? And then, so then they'll click on this, and then all of their sites will show up, and then they'll click on the site that they want, that's submitting or whatever, and then it'll pull all the correct information in about that site. Okay? And then over time, that the site information that the submitter is seeing here, if they're using the web submission tool, is the exact same tables that our clerical staff is seeing if they're getting it in on paper. So there'll be a way, kind of a QA/QC over time, of these sites went away, we got new sites, we need to add them, all that for going forward. Okay? And then again, the test selection tools and so on look the same. So again, kind of a stepwise process, really all aimed at these things to say is how do we improve the quality, especially around this identity preservation and the value of the diagnostic information. And really our ultimate goal here is to say, how are we better positioning ourselves to support the need of kind of this, what I call next generation of food animal agriculture and production animal medicine going forward. And these, I wanted to acknowledge these folks. These are people that actually do the work. A couple of really sharp, talented young women. And we have Katie. She's really new. She just started this spring, 2014 grad, and she's helping reach out to the clients to get these things set up, right?
And then Kate has been with us for about 2 years now, and she's really been the one working on the diagnostic database side of things. I did want to bring this up too and just answer any questions people have for what we know. I tell you, we really don't hear much about this since it's kind of, so it would be interested just in conversation today, throughout the day, is like, what are the clients seeing, feeling, practitioners, what kind of interaction is occurring? Because we're not really privy to much of any of that. But I did want to say, kind of what our role is in this as it relates to helping our clients comply with this federal order. And really our goal in it is to say, how do we streamline it as much as possible for our clients and make it as easy and painless as it is for possible? Because essentially what we're doing is, for this, since this came into place, is we're doing the tests, we report the results back to our clients like we always have, and then the difference is here that the PEDv and the Delta coronavirus are going to the USDA, and then if they're positive sites, as I understand it, then they're calling the practitioner to get additional information, seeing if there's a need for a herd plan and so on. And again, this part of it is, you guys know a lot more about that than I do, because that we're not really privy to that, so don't really have much of a feel for that at all.